Hello and welcome. This is the Bio 103 Lab Final Review. Okay, Lab Final Review. Now, before I go anywhere, it's very important that you go to Canvas, you go into our modules, you go down to the Lab Final area of the modules, and you view the study guide for this exam. Okay, you can find it there, very straightforward, it's not that long. You can go in and find all of the study materials that I've provided for you. I'm here to tell you, if you can go through the handouts that I've made for you, the worksheets for each lab, and you can answer all of those questions without a whole lot of effort, your chance of getting a high score on this test will be dramatically elevated. I see zero chance that you could be going over your study guide, checking on the worksheets, and listening to this conversation and having any capacity to do poorly on your exam, okay? So as long as you're working, you should do okay, all right? But make sure that you understand the difference between looking at a question, looking up the answer, and kind of remembering it as opposed to learning what it means. Okay, so with that in mind, I have broken up my sheet of paper here in front of me into the individual labs which we covered. So I've got a little section on enzymes, I've got a little section on photosynthesis and respiration, that will be the fermentation stuff we did. I have a little area on mitosis and DNA, and then at the end I have a little bit of an area on taxonomy. Okay, enzymes, photosynthesis and respiration, mitosis and DNA, and taxonomy. Now, as I look at my sheet of paper, I'm here to tell you I got 2 lines of text about enzymes, and that's it. I got 2 lines of text about taxonomy, and that's it. And the whole rest of this is photosynthesis, respiration, mitosis, and DNA. All right? So you can expect a lot of questions on those middle labs as a result of this. All right? And I'm going to be very out front with you. I'm going to tell you exactly what's coming, so prepare yourselves. All right. First, on enzymes. Now, the enzyme that we used in lab was catalase. We used a catalase experiment. That's catalase that we pulled from animal material mostly. In other words, it came out of liver. I believe it was chicken liver. All right, so we used liver tissue, which contains the enzyme catalase, to break down hydrogen peroxide. You need to know what the reactants, the substrate, the reactants going into this catalyzed reaction are. You need to know what the products are that come out of it. Okay, we got peroxide going in. What is the resulting products from an enzyme catalyzed reaction using catalase on peroxide? Is catalase destroyed in the reaction? Tell me about, we use different pHs. Why did we use different pHs? What did that tell us? We use different temperature regimes when running catalase experiments. What did that tell us? What happens if the what happens if the temperature of the substrate of the reactants gets too low? What happens if it gets too high? What if it gets super high? What does that do to catalase, which, mind you, is a protein? That's a hint. Okay, this is very important. So you need to understand the concepts of catalase and catalase reactions. Okay, moving on to photosynthesis and respiration. First things first, let's talk about plants and photosynthesis. You need to understand the process of chromatography. What did we put in our chromatography apparatuses? All right, tell me about this paper. What did we put on that paper? When we ran fluid through said paper, what happened to the pigments that were rising there? Okay, what are those pigments? Are some of those pigments more important than others? Why do they represent certain colorations? When I look at xanthophyll, why does it have a certain color as compared to chlorophyll A or B? Okay, so you need to be able to tell me about this. Now, in a leaf, you need to be able to walk me through a leaf model. Tell me about the top and bottom surfaces. Tell me about the different like mesophyll within there. What's a stomate? What are guard cells? What are they for? Okay, I want to know about all this stuff, man. You got to know it. All right, inside of that leaf, be able to tell me about chloroplast. What do chloroplasts do for the plant? What are the parts inside of the chloroplast, and what are they responsible for? All right. Let's see, what else do I have to say here? Yeah, parts inside the chloroplast. What happens where in terms of photosynthesis? Yeah, man, I want to know all about it. You need to understand the concept. Card cells, yeah. Mesophyll. Okay. On to respiration. I want to know what a respirometer is. Okay. What is this two-tube system that we utilized and referred to it as a respirometer? This is actually a super important tool for measuring fermentation reactions in microbiological laboratories. Do this every day in a micro lab looking for fermentation. What gas is being produced here versus in a catalase reaction? Okay. What organism is being used here in a fermentative reaction like we did in lab as compared to what we did with catalase? All right. This is a very different situation. In what ways do you as a human utilize potentially fermentation? In what ways do we as a society utilize fermentation in the world around us? So this is very important. It's a very important concept. Are there ups and downs to fermentative reactions versus standard cellular respiration? I want to know about it. All right. Yeah. Okay. Mitosis and DNA, changing gears. I want to know, first things first. All right, let's just get this out of the way. I want to know every step in DNA extraction. I want to know what's done, and I want to know why it's done. Because I think it's just a wonderful biological experience to grasp that you can bust up a cell, but it's going to be phospholipids, and it's going to close back up. All right, that cell's going to reseal itself, kind of like with endocytosis and exocytosis. But you have to take steps when you do a DNA extraction to prevent the cell from reforming. What do we use to prevent the cell from reforming? Once all the internal parts and pieces of that cell are busted up, how do we keep from breaking down the DNA that's released from the nuclei that are there? All right? What do we apply to this to prevent that? Tell me about all the steps, man. Because if you can tell me about those steps to extracting DNA, man, that's really going to show that you understand some science. All right? It's a really good story to tell. So you need to know about the steps involved in DNA extraction from like a banana, for example, what it looks like, and the why of it all. Like, how does it all work? Okay? Let's see. All right. You obviously need to know the stages of mitosis, okay? From an image, I'm going to show you micrographs. You got to tell me if it's, prophase, metaphase, anaphase, what have you. On this, you're going to have to tell me if it's in kingdom Animalia or kingdom Plantae. I want to know if it's an animal or a plant. It's very obvious. Plants have square cells and big honking chromosomes. Animal cells tend to have kind of round margins, and they have smaller, harder to see chromosomes. So it should be very telling. If it's obvious, probably a plant. If it's requiring you to look a little bit, probably an animal, okay? Be able to tell the difference. Understand the difference between mitosis and meiosis. Also in that lab, you looked at seminiferous tubules, and you looked at ovary slides. In that seminiferous tubule, one, you got to be able to tell me that comes from the testis. You got to tell me they're seminiferous tubules. Pointing out the spermatids or the sperm tails would be a good idea. Pointing out the Leydig cells and telling me about testosterone would be a good idea. You need to know the parts and pieces. Understanding that those spermatids are haploid. All right? Haploid means they have half the normal cellular nucleic component, half of the DNA. As opposed to any cell going through mitosis that would be diploid. Like if I look at my arm right now and I look at my skin's made up of diploid cells. But sperm and egg, those are haploid. Right? Those are haploid. If you don't understand that, we can talk about it in a second. On an ovary slide, you need to be able to notice that there are different types of follicles. Not necessarily naming the different types of follicles, but understanding that they exist. Okay, that there are indeed different types of follicles. Primordial, primary, these are good things to know. Inside of those follicles, you will often see an oocyte. That is the physical egg in the ovary preparing for maturation. You need to grasp that much like a sperm, an egg is haploid. Okay, and those are found inside of those follicles. You might need to know a little bit about what a corpus luteum is and what it does. Like, these are important concepts for the ovary. So being able to look at an ovary, and prepare yourselves for, well, never mind. Let me just not go there. I've received some interesting answers over the years for some of these questions. Okay, so testis and ovary, parts and pieces within and what they do. Yep, that'll work. In mitosis, we talked about the different stages, animal versus plant. It's a worthy idea to know how they divide terminally. So at the end of the day, how do they go through cytokinesis? Animals and plants do this in different ways. You should know the different ways in which they do this. Let's see. And I think that moves us on down to taxonomy. So let's do taxonomy. Last but not least. All right. First things first. The vast majority of what I'm going to ask you about taxonomy is going to be pretty hard to screw up if you paid attention to the quiz and the worksheet I provided you. All right? Pretty hard to screw it up. You need to make sure that you can use the key that I provided in those PowerPoints and stuff that I put up on the Canvas page. Okay? If you can walk through that key and you understand how it's utilized, you're going to be in good shape on most of these questions. Because all I'm asking is phylum and class for the most part. So you got to be able to look at it, go through the key. I will provide the key for you. Tell me what the phylum is. Tell me what the class is. And here's the only nuance to this that makes it mildly complicated. You got to tell me the common name of the organism as well. Okay? So you can tell me it's a Hyrudinian, but at the end of the day, you also got to tell me that's a leech. You need to be able to tell me it's a crustacean, but at the end of the day, you got to be able to tell me it's a crawfish or a crayfish. All right? I don't need some sort of really ridiculous fancy naming methodology. If it's a snail, it's a snail. Okay? I don't need Eastern Hercules beetle. I need beetle. Okay? Nothing fancy. Nothing fancy. So the key that I provided you, just make sure you can go through it. And the organisms that I distinctly talked about in lab, the images that I provided you, it's a good idea to know their common names. All right, and that's like 8 or 9 questions right there. It's going to be phylum, class, and common name. That's, what, 18 points or so? That's a lot of points on your test, okay? A huge percentage of your test. Which is freebies from my perspective. Like, you should just know these. All right, next, let's see. You need to know the domains. So tell me about what a prokaryote and what a eukaryote is. In the eukaryotic kingdoms, okay, you need to know the eukaryotic kingdoms. Tell me kind of what it takes to be a protist. Tell me what it takes to be an animal, to be a plant. All right, I need a little bit of information, a little bit of detail about what it takes to get in there. All right. Tell me about Linnaean taxonomy. I thought I had this in here someplace, but I swear I don't see it. Anyway, tell me about Linnaean taxonomy. I want to know about binomial nomenclature. Okay? You got to be able to tell me how you would write that out. For instance, you are a Homo sapien, yeah? Or how about, let's do something random. Felis concolor. That's a panther. Okay? Felis concolor. Now, the scientific name is Felis concolor. That's a genus and a species. All right? Genus, Felis, species, concolor. The scientific name is Felis concolor. But its common name is a panther. Okay? It's just a panther. Now, if I'm writing this out in a text of some form, I'm going to italicize Felis concolor. I'm going to capitalize the genus name. So capital F for Felis, lowercase c for concolor. Felis concolor, italicized, capital C, or F, capital F. You capitalize the genus, lowercase the species. When you see capital genus, lowercase species, and it's in italics in writing, you know that's a species name. All right? This is very important. It's Linnaean taxonomy. This is how we classify organisms. The end. Okay? So that is about the extent of review that I'm going to provide at this stage. Again, with this, with the study guide, with the worksheets and the quizzes, zero chance you should fail this. You should do extraordinarily well. You should do fantastic. Get to work. I hope you just knock this out of the park. Nothing would make me happier than a bunch of high grades. But mind you, that the first grade you receive will be quite low, because I have to go through and apply points to them, because this is a fill-in-the-blank test. Okay? So expect a low first grade, and then I'll go through and fix it over the next few days. Or after the test, that is. All right, it's 50 questions, and it's going to be fill-in-the-blank, it's going to be honor lock, it's going to be timed at like 55 minutes to an hour, so be ready. All right, thanks, guys.